All right. Welcome everyone to Quantitative Seminar. I'm Angela, this is Connor, we're your coordinators for this year, and I'm going to start with our land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. Today we have Roger Peters joining us. He is a supervisory fish and wildlife biologist with the Washington Fish and Wildlife Conservation Office of the U.S. Fish and Wildlife Service in Lacey, Washington. He's worked for Fish and Wildlife Service since 1993, received his PhD from the University of Washington School of Fisheries in 1996. Roger is a Squaxin Island tribal member and enjoys fishing, hunting, hiking, and spending time with his family. Roger will take questions at any time during the presentation, so please join me in welcoming Roger.
Well, thanks everybody for attending the presentation on a Friday afternoon. Probably not the most desirable activity today. Like Angela said, I'll be talking about life history diversity in the Elwha River during and following dam removal. I want to acknowledge the large list of co-authors on this project. This was a really collaborative project and great group to work with. I really want to acknowledge George Pest. George had the foresight of making sure samples were collected, being able to get the samples processed, which was at the time was really difficult because it's kind of an add-on to the overall project that we were working on. I'd also like to acknowledge all the agencies that allowed their staff to contribute to this project. Also, all my Elwha talks, I acknowledge the larger Elwha community. This slide doesn't do justice to all the folks that worked on this Elwha project. An extremely collaborative group to work with, some brilliant people. It's been a joy, probably the highlight of my career to work with these folks. But they've really been a great group to work with. For the talk today, it's based on a paper that we've recently published in Frontiers in Ecology and Evolution. It's a special issue on intensively monitored watersheds. So I'm going to talk a little bit about intensively monitored watersheds, what they are, what they do, provide some background information for the Elwha project and for this project specifically, go into methods, the results, summaries, and then I have a recommendation slide as well. So intensively monitored watersheds are essentially long-term monitoring of one or more catchments. Examples include the Alsea and Carnation Creek watersheds during the late 1970s, early 1980s, essentially to assess the impacts of logging on watershed ecosystems. Now, more recently, there's a series of 19 intensively monitored watersheds looking at the impacts or benefits of restoration throughout the Pacific Northwest. And the Elwha Dam Removal Project is another example of that. IMWs provide valuable information on salmon ecology. They also let us understand the connection between watershed geophysical and ecological processes and functions. And kind of the big one that jumped out to me was this delayed effects identification. So from the Carnation Creek study, the first 30 years of that study basically suggested that logging was good for coho salmon. We had warmer streams, juvenile coho grew better freshwater survival, and smolt production was up about 33%. About in the late 1980s, there was a landslide in the basin due to logging. It took about 20 years for that landslide to translate through the basin. After that, we saw reduced fry to smolt survival and reduced smolt production to about 60% of the original pre-logged production in the basin, or 33% of the peak during the thermal benefits of logging. So this kind of allows us to understand these legacy effects of land management impacts and the discussion of sliding baselines. So I think from that standpoint, they're really critical, to have a 50-year baseline to look back on and kind of identify all the factors and how they may change in ways that we may not have predicted. The Elwha Basin, it's located on the Olympic Peninsula of Washington State. It drains the north slope of the Olympic Mountains and is predominantly within Olympic National Park. In the early 1900s, 2 dams were built. Neither of the dams had fish passage, so they effectively blocked 90% of the watershed to anadromous fish, and also limited migrations of resident fish such as bull trout or mykiss to basically downstream and dropping over the dams and then being locked in a new reach or within the reach where they were located. Elwha Dam was built in 1912 at about river kilometer 8. Glines Canyon Dam was built in 1927 at river kilometer 21. In September of 2011, dam removal was initiated for both of the structures, with Elwha Dam being removed by the spring of 2011 and Glines Canyon Dam being removed in September of 2014. Unfortunately, November of 2014, during a storm, there was a rock fall off the canyon downstream of Glines Canyon Dam, which again blocked fish migration, and that was subsequently removed during the summer of 2015 and 2016. But all of that delayed migration into the upper watershed. So the management actions in the Elwha River, there's basically 4 management actions that either have potential to influence mykiss life history or allowed us to monitor changes in life history. Dam removal projects that I just talked about, but also there was hatchery modifications. The Lower Elwha Clallam tribe operated a production facility rearing Chambers Creek winter steelhead, so a non-native stock. That was converted in 2011 to a conservation hatchery rearing Elwha native stock that was basically removed from, well, they just made pumping to get eggs, developed a captive brood stock to develop that program. There's also an in-river fishing moratorium that was initiated in 2011 to essentially protect these fish during what was suspected to be a major impact period with all this sediment being released from these 2 dams. It's still in effect today, except for coho salmon. In 2023, the Lower Elwha Clallam tribe initiated a ceremonial subsistence fishery for coho. They're targeting 400 fish. They've yet to catch 400 fish in those fisheries. And then finally, the National Marine Fisheries required the Olympic National Park, who was the project sponsor, in their buyout, they required them to develop a monitoring adaptive management plan and provide 10 years of funding to monitor the impacts of dam removal, essentially to make sure that dam removal did not cause the extinction of Elwha steelhead and Chinook. So it's that monitoring that has allowed us to do this project plus significantly more monitoring within the watershed. That monitoring adaptive management plan was focused on steelhead and Chinook. And it was so focused on viable salmon population metrics. So this shows an example of the metrics for winter steelhead. So running from top to bottom, you have entry timing, adult abundance, smolt per female, adult to adult productivity, and then spatial extent. And you can see that, so the x-axis is year, this blue line represents when dam removal was initiated. The blue dots are annual estimates, and the brown circles or lines represent 4-year geometric means. And that was the metric that we used in the monitoring adaptive management plan to determine if we had met triggers to move to a different level of recovery. And you see that entry timing has basically been consistent through time. Adult abundance has increased, smolt per female has increased. Adult to adult productivity has been kind of variable, but it's met the trigger value that we thought we needed to have population growth. And spatial extent has increased over time as well. So given these, one question might be, well, why are you interested in looking at life history since the population looks like it's doing so well? And the reason is that life history's traits influence fitness. They're subject to selection pressures, and so they allow stocks to basically adapt to the conditions within the watershed. Also, the spatial and temporal differences, diversification spreads the risk to the population. So it reduces the risk of extirpation through portfolio effects, as described by Daniel Schindler. And these asynchronous life history and environmental conditions have been shown to mediate declines in Chinook and steelhead. Life history traits are also influenced by several biological and physical factors, and these will interact with each other. For example, temperature will interact with physiology and productivity that'll influence growth that thereby can influence life history strategies. And the Elwha River has a very diverse physical environment, from headwaters that are very cold, snowbound, to lower elevation areas more subject to winter floods, etc. In addition, mykiss are known to express a significant, really diverse life history patterns. More than 30 life histories are common within a single watershed. They display partial migration, where some fish will migrate, some fish will not. The fish that migrate can have variable return timing, either returning in the summer or winter. And a lot of this life history variation is due to variations in freshwater and marine residency, total age, and repeat spawning. So the objectives of our study were to look at these 4 management activities and go, how does mykiss life history increase through time, potentially in relation to these three primary management activities? And will that diversion be due to altered migration patterns or timing, differences in freshwater or marine residency, or degree of repeat spawning? And then also wanted to assess whether or not changes were associated with gene flow from within or outside the Elwha Basin. So on to the methods. Life history diversity can be defined in a number of ways. We defined it basically as migration strategy. Do you migrate or not? If you migrate, what's your run timing? And then how long do you spend in freshwater phase, marine phase, or what's your total age? And then what's the degree of repeat spawning? Sample collection was opportunistic through the rest of the more focused monitoring activities. And this actually comes back to bite us a little, and I'll talk about that in a little bit. But we collected samples during species composition sampling for sonar estimates to estimate winter steelhead abundance. That was led by Keith Denton. Telemetry studies to assess distribution of adults early on in the project, led by Sam Brinkman. Our assessment to determine what the proportion of hatchery-origin winter steelhead was in the steelhead population. And then my coworker, Jeff Johnson's assessment of encounter rates. If we initiate a recreational fishery back in the park, what will the encounter rate be for mykiss and for bull trout? Once we had fish, we identified their origin, measured their length, noted their sex. We also classified them as a new entry, a holding fish, or a spawn fish, and that kind of helped us assign them to summer or winter runs. And then we collected scales and fin clips for aging and genetic analysis. The scales were read by Andrew Claiborne at WDFW Aging Lab, and they use the European age notation to describe those fish. And I'm going to provide a quick example here because I have a couple of slides that will show that. A simple number represents resident fish, so a 3 means that it was a resident fish and it was 3 years old. Summer runs are identified by a number, which is how long they were in freshwater, a period showing that they migrated, and another number showing how long they were in marine environment. Winter steelhead are the same, except for they have a plus at the end of them to note them from summer steelhead. And then at the end of all of these, you can have an S or a series of S's, or an S and a number and an S, and that signifies repeat spawn. So double S's mean that fish has returned to spawn 2 times. It did it in consecutive years, and this is its 3rd spawning migration. The S1S means that it spawned once, it took a year off, an extra year in the ocean, came back and spawned again.
So can you show me on the box what a spawning scar looks like? Because I'm used to looking at sockeye, which they just spawn and they die.
So these are spawning scars right here, and right here.
And that's from one spawning event?
So these are 3 successive spawning events. So you're going to have limited growth. Once the fish is an adult and come back, as they go back to the ocean, they don't really grow as much as you would see between these other periods, because a lot of their time is spent reconditioning and getting themselves backed up to, back into condition after spending so much energy to spawn.
Huh. So that would be 3 separate years.
Yep.
Is there a limit to how many spawning events they can have?
3. I think it's been 5 or 6 repeat spawning activities have been observed. So it's a limit of how many times you can avoid predators, how many times you can recover. But yeah, 3 is not an uncommon value. Then genetics. This work was led by Alex Frake, Kristen Nichols, and Daniel Pierce. And their designs differed a little bit from ours. They actually had sufficient number of samples prior to dam removal to actually compare pre, during, and post. They used they generated rat sequence data using best rat protocol. They excluded fish with O'Clarke ancestry, and they used program admixture to identify the number of genetic clusters within the basin, the frequency of each genetic cluster ancestry, the sum of total ancestry, and the proportion of ancestry admixture, or mixture, I'm sorry. And then statistics. Basically, we broke our data set up into 2 time periods: during dam removal, 2013 to 2018, and post dam removal, 2019 to 2024. And we did that because we didn't have enough samples during some years to actually just run the data through a continuous analysis. We used the CMH test to compare during to post dam removal, but also to compare hatchery origin fish to natural origin fish. And throughout the presentation, I have hatchery origin fish as HORs, natural origin fish as NORs. And in some of the slides, you'll actually see the river broke up into sections. So the lower river is from the mouth up to Elwha Dam. The middle section is between the 2 dams, and the upper section is upstream of Glines Canyon Dam. So on to the results. This slide basically shows the distribution of our samples. The circles represent during dam removal, the triangles post. Dark colors are few samples, light colors are more samples. And then this is the zoom in of the lower river. And you can see that most of our samples were collected in the lower river during Keith's species composition sampling for sonar. That's where we needed the most intensive sampling. And so that's where most of our samples came from. As you move into the middle river, you see we have very few samples during dam removal, more sampling locations, and more samples after dam removal. And in the upper river, we had a single sampling location with less than 10 samples. Prior to dam removal, we did a better job and got out, and this was during some of Jeff's work, getting more fish after dam removal, including up into the extreme part of the basin. And this is where that kind of comes back to bite us, this different unbalanced sampling. So this chart shows unique life histories or just life histories in some cases. By river section, during is in red, blue is post. The size of these circles represent how many samples were collected. We have all mykiss, resident mykiss, summer steelhead, and winter steelhead. Where the circle is relative to the y-axis is how many life histories were observed. So these 300 samples in lower river represent about 20 life histories. The bars represent how many unique life histories that were observed for that sample. And you can see as you move up river, we start seeing more and more diversity with fewer and fewer samples. So the fact that we have increasing diversity as you move upstream, and we sampled more upstream after dam removal, impacts our ability to really say how these management actions influence life history diversity. And that was one of the things of, we knew we were going to do this, but we're collecting data in so many different ways over such a long time period that none of us were really kind of monitoring where our samples came from. We knew we were sampling in the upper middle area prior to dam removal, but through time, obviously didn't see that we had this difference, and that was a major impact of this study, quite frankly. It was really disappointing when it came to light, like, oh, that's unfortunate. So even when you're in an intensively monitored watershed, every aspect of what you're going to do needs to be pushed through an experimental design before before proceeding too much.
But I think you still have quite a bit of information there, though. I mean,
Oh, yeah.
back to that slide. So in the post period, like if you go back one, obviously in the post period you have fewer samples, the blue ones.
So
fewer samples but more life history strategy. So clearly that increases with going from lower to middle to upper.
Well, we see we have relative to before dam removal, we have many more samples. So had we collected more samples up here, there's theoretically this could be up there. I don't believe that it would be, but if you look at the numbers, you can explain some of the differences just by the fact that we had more samples and therefore more life histories.
Maybe you could do like a resampling analysis though, where you could take the upper post data and say, what if you I don't know how many samples there are in upper during, let's say that's 10. You could say, what if you resampled, randomly took out 10 of the post ones how many life history strategies are there, repeat, take out 10, how many life histories are there, keep repeating, and you get a distribution of expected life histories you can compare with the actual one you observed and say that more or less than you'd expect.
We actually did that. Martin Lehrman did a lot of the statistics on this. He actually ran that analysis and it actually shows a difference. But in some discussions and debate, there were some of the assumptions that are a bit violated from that analysis. So we didn't feel comfortable leaving it in the paper. And some of the other results were implied basically the same thing. So we were comfortable leaving that out, being more conservative than our proclamations of what had actually happened based on this sampling impact.
Okay.
Maybe a curious question on that too, like even though you have more samples in the post dam removal versus the during dam removal, like is there a way you could like just like standardize each of them, so like you would divide, I don't know, the number of unique life histories that you found in each time period by the number of samples?
You can't, that's what one of the, so actually this, in the paper, there's actually another panel to this, where Jeff actually looked at based on how many unique life histories or how many life histories are observed per sample size. And it's still influenced by the same thing where you can have, you know, you'll still see the same pattern as your sample size increases because you start replicating the same sample over time, that artificially goes down. So,
so limit to how many life histories there are.
Yeah.
I mean, sample like 40,000 times, you'd think it was a number per sample, but it's
exactly.
Really, it's a bit like sampling how many unique species are there. You get the species curves. I think the more times you sample, eventually you get the right number of total species.
So, and this will kind of point to some of that. So this is a slide that Andrew Claiborne helped me put together. Basically, here's that European notation of age class year. The dashed line represents the during and post period. The circle represents the proportion of fish that displayed each life history pattern, and the color represents age. And so you can see that a large proportion of the fish were specific age classes, and there's a lot that were very uncommon. So you get, it's harder and harder to get those samples through time. But from our study, we identified 39 different life history strategies when we combined HOR and NOR mykiss. There were 18 during dam removal, 38 after dam removal. And hatchery origin fish were much less diverse than natural origin fish. During dam removal, they displayed 5 life history strategies where NORs displayed 17. After dam removal, hatchery origin fish displayed 9 and natural origin fish displayed 36. We had both anadromous and resident fish, and the anadromous fish had a summer and winter run timing. And that's important. During our riverscape surveys in 2007 and 2008, we snorkeled the entire river, and in the lower river, we didn't observe a single summer steelhead during those 2 years. Other surveys were completed. Basically, folks thought summer steelhead were extirpated in the system. So that's a pretty amazing recovery that 3 generations later, we have summer steelhead, and in some years, estimates were up to maybe 900 summer steelhead in the basin, which would essentially mean there's more summer run steelhead in the Elwha River 12 years after dam removal than there are on any of the other Olympic Peninsula rivers.
So what's the difference between 2.1 and 2.1 plus? I'm not sure. I'm not familiar with the notation.
So that's what I went over. So it's 2 years in freshwater and a year in saltwater or in the marine environment.
But below it, that's 2.1. So what's the difference between 2.1 plus and 2.1?
So that's a summer steelhead there. The 2.1 is a summer steelhead.
And 2.1 plus is winter?
Is a winter steelhead, yeah.
Oh, I see.
And what's like 1 plus mean? Like
so that was actually a fish that had gone out, spent a year in freshwater, looked like it had gone to sea, but probably only spent a portion of the time.
Okay, interesting. Okay.
And we actually saw some fish that were like that. It was going to be a part of the paper. We crunched that out to make the paper smaller, where we looked at mid-sized mykiss, 450 to 550 millimeters, and did some chemistry analysis on those, and basically about 40% of those fish are actually anadromous. So This is the same slide, except for it's winter steelhead, hatchery origin fish, and natural origin fish. We observed 20 different life history strategies, 11 during and 20 post dam removal. Again, HORs were displayed less life history diversity than NORs. In fact, they did not increase after dam removal like mykiss combined did. Where NORs went from 11 to 19 to 19 life history strategies after dam removal. So I'm going to show a series of 4 charts here, and these are all for winter steelhead. I have proportion on the y-axis. Again, red is during dam removal, blue is after dam removal. And then the x-axis is going to change, and this one is how long they spent freshwater or their outmigration age. And basically, fish were primarily age 2 when they left the freshwater. It varied by period, and it also varied by origin. Natural origin fish were older after dam removal. You can see that you have a pretty significant reduction in age 1 fish, an increase in age 2, and then you also see some age 4 outmigrants in the watershed. Natural origin varied from hatchery origin during both periods, and that's largely because all the hatchery fish are released at age 2. So this is years at sea, essentially the same results, primarily 2 years at sea, varied again by origin and period. And again, natural origin fish spent more time at sea after dam removal. You can see again a reduction in age 1, a much greater increase in age 2, and then you all again start seeing some age 4 fish. Natural origin fish varied from hatchery origin fish during dam removal. You can see that actually post dam removal, the blue bars, natural origin fish actually kind of shifted to be more like the hatchery origin fish in their marine migration patterns. Total age, primarily age 4, again, varied by period and origin. Natural origin fish were older than during the post dam removal period, and that makes sense. If you're older when you leave to go to freshwater, and you spend more time in the marine environment, you're going to be older as a total age. And natural origin fish were older than hatchery origin fish during both periods. Now this is repeat spawning, varied by origin. With hatchery origin fish, natural origin fish displaying greater repeat spawning life history after dam removal. Natural origin fish were about 2.9% repeat spawning during dam removal, 10.2% after. Hatchery origin fish less, 0.6% and 3.8%. And you can see that hatchery origin fish displayed a single repeat life history strategy. And in contrast, natural origin fish displayed 5 different repeat spawning life history strategies. They repeat spawn in consecutive years, as identified by those. So we have documentation of fish repeat spawning at least 3 times. They also took a year off, as in these 2 cases, before coming back to spawn. This again, so pretty diverse. I'm going to talk about some results for summer steelhead and resident fish. These are really general. We had about 50 samples for resident fish and less than 50 samples for summer steelhead. Summer steelhead were primarily age 5, so they're a bit older than winter steelhead. They're primarily 2.2 age class, and they repeat spawn about 6.9% of the time. Resident fish, I broke these out by hatchery and natural origin. Again, hatchery origin is red here and natural origin is in blue. Even though we only had 8 hatchery origin fish, you can see that they're primarily age 3 to 5. Hatchery origin fish displayed 6 life history strategies, natural origin 12. Significantly more repeat spawning in resident fish than there were in either of the anadromous counterparts, so about 20%. So twice of what the winter steelhead were doing, nearly 3 times what summer steelhead were doing. These are the genetic results from Alex and others. So the first y-axis is ancestry coefficient. Second y-axis is observed heterozygosity. The x-axis is basically time or years. You broke it out in different strategies. Green is middle river, brown is upper, Purple is lower, red is south branch of Little River. The dashed line here represents admixture coefficient. And the solid line represents observed heterozygosity. And basically there are 4 genetic clusters in the Elwha River. There was no new genetic clusters that developed during or after dam removal. However, there was significant ancestry, significant change in ancestry through time. Before dam removal, it was primarily lower river, and then we saw observed significant increases in admixture. After dam removal, it was primarily the upriver genetic cluster upstream of lower Rica Canyon, I'm sorry. And all of this stuff taken together suggests that the gene flow was primarily from within the Elwha River. Well, there was a slight increase in heterozygosity during dam removal and a slight decrease during post-dam removal relative to before dam removal, but this is largely likely due to the decreased sample size, where pre-dam removal we had 752 samples, post-dam removal there was 178. Natural origin fish increased in upriver ancestry over time, and hatchery origin fish basically remained lower river ancestry, and it did not change over time. That's likely due to the fact that most of the hatchery fish stayed in lower river. Very few hatchery fish, based on our sampling, had really ascended upstream of Elwha Dam. And there's a positive correlation on the frequency of admixture events after dam removal for natural origin fish and not for hatchery origin fish. So summary, the IMWs allowed us to document the combined positive influence of the management actions. We identified 39 life history patterns in this 12-year study, basically 3 generations of mykiss. So that's similar to much larger watersheds such as a Klamath. There was 18 during dam removal, 38 post-dam removal. Unfortunately, our sampling, relying on just getting samples through other monitoring activities, impacted our ability to really strongly say that that was due to the management actions in the Elwha. We identified resident and migratory mykiss, and the migratory individuals displayed summer and winter return timing. It also allowed us to identify changes in freshwater and marine residency for winter steelhead, their total age, changes in repeat spawning, and to note that these are likely due to period and to origin. Life history diversity increased substantially more in winter or NOR fish than it did in hatchery origin fish. Genetic structure decreased, diversity was unchanged, and potentially it's lagging behind life history diversity. With the increased time it took to get Elwha Dam or Glines Canyon Dam out, and then the subsequent rockfall, reduced fish getting up there for quite some time. And also we're sampling, perhaps the progeny of the fish that we sampled at the end of our study will actually show that genetic diversity increase. And it doesn't appear that there was any immigration from outside the Elwha River. And the recommendations, I'd recommend continuing to do this type of monitoring in the Elwha River. But before doing so, going through some of the processes that you talked about, we need to develop this experimental design so we end up with more balanced samples throughout time. And we recommend the continued sampling because it's very unlikely that the basin has reached equilibrium. In addition, hopefully at some point, steelhead will recover to a point where people are fishing on them. And so it really provides a unique opportunity for us to go, okay, here's life history diversity, we enact fishing. How does that influence life history diversity? Do we start seeing impacts on repeat spawning? Andrew Claiborne published a paper a couple months before we published this, and basically showed at one point in the Hoh River, which is just around to the west side of the Olympic Peninsula, up to I can't remember the years off the top of my head, but I think it was the mid-1980s, up to 63% of the life histories observed were repeat spawning life histories. And repeat spawners are often thought to be more productive than first-time spawners and actually help with more with population growth than the first-time spawners. And with that, I'm done. And again, I thank you for attending the presentation. And I'm happy to take any additional questions.
Yeah, thank you, Dr. Peters, and anyone on the Zoom. If you have any questions, please just feel free to type it into the Zoom chat or unmute your mic.
How many folks on Zoom?
We've got 12 people.
It's usually a big crowd on Zoom.
Yeah.
Just in case you're wondering at the mixture of people listening and listening.
Yeah, we should. I do have a question.
Yep.
So I was really interested in the much more diverse life history of natural origin fish. Why do you think of that? Would you think it's because the hatchery origin fish are less genetically diverse, or because of some kind of an adaptation we know that they go through during agriculture?
I think it's the fact that they're all released at age 2. So a lot of life history determinations happen in the juvenile stage. Steelhead really documented well for Atlantic salmon, but I think it's also been documented, and the two species are so close that you'll see a lot of life history divergence determined at age 1 for juvenile Atlantic salmon, where they'll decide I'm gonna migrate, or I'm not gonna migrate this year. I'm gonna hold off, and then they return at different rates. And so I think it's the fact that you truncate them you're going out at age 2. And when you go out at age 2, that influences what they're gonna do for the rest of their life. So really, it'd be interesting to see someone have an open hatchery environment where fish can actually leave as they want, and see if that influences life history diversity.
Thank you. It's interesting, it is an issue.
Yeah, is it common, what is the release date for other salmon?
Predominantly age, well, for steelhead, for most of the production facilities, it's age 1. They can grow them in the hatchery fast enough to produce them and let them go at age 1 and have good survivals. In fact, that's what the Lower Elwha tribe did with the Chambers Creek stock. It was just production facility, get them big, get them out. And then you don't have to hold them as long, it's not as much food. But when they converted to a conservation hatchery, like, okay, most of these fish are going out as age 2. We'll try to replicate that and be as consistent as we can with what's happening in the natural environment. And predominantly they were, because most of the fish do that. But there are the segments that go out as 1 or 2 or 3 or 4 that you do not see in the hatchery environment. And I think that's where that diverges.
What do they do for pink salmon? Is pink salmon just like a couple of months?
Yeah, pink salmon are really fast. Yeah, they emerge. I think it's within a month. We did some pink salmon stuff on the Elwha early on, and in discussing those with folks that were experiencing that, it's very time dependent on how successful the program will be when you get them out of the facility.
What's the ratio of the different salmonid species in that Elwha system? Is it pink, or is it mostly?
Well, historically it was mostly pink. Currently, it's probably Chinook would be about Chinook and coho would be about even at 4,000 to 5,000 fish. Steelhead would be next with 1,500. Actually, this last year, the pink population has increased over the last 10 years or so, from about 600 or so up to 35,000 last year. So they are starting to actually increase. So now they are actually the dominant species, but before that they were not. Chum salmon are also, we're starting to see more and more of those, but we don't have a population estimate for those.
I was gonna say, pinks are pretty the most adaptive to like figuring out this new habitat and taking advantage of it. Yeah, but they were at such low numbers during dam removal that there was a few hundred pink salmon in the Elwha before dam removal. The substrate was really large. There was a few side channels that they could use, but even those had relatively coarse substrate, just because all the substrate was blocked by the Elwha and landscaping dam. Good questions on that?
Not seeing any questions.
I've just been struck by the invasion of pinks in Norway. I'm sure you've heard of that.
Yeah.
Which I believe was like a Russian program where we're gonna bring pink salmon in and see what happens.
Yeah, I think.
And see if they establish themselves, and then they did. And I mean, I've just seen some staggering. They built a weir across this one river, and they were like like hundreds of thousands or millions of pink salmon in this tiny little river.
Yeah.
It's just insane
Mm.
numbers.
Mm.
And they're like, well, these aren't native, we should get rid of them. I'm like, hate to tell you, but it might be too late.
Yeah.
Are there any upcoming like, so I know like the dam removal was the big like event like that obviously has like a lot of like ecological effects. I don't know. Are there any upcoming projects that like are planning to be done on the river? I just asked because back in my hometown in Monterey, California, there was the San Clemente Dam removal there. And so like in the beginning, it was like, okay, we're going to just like remove the dam and call it a day. And that's what they did. But then after that, they're like, actually, we need to restore this place. And so like I think a lot of time and effort like went to like sort of restoring and like trying to make sure the habitat of that post dam removal was like suitable for fish populations, for just benthic macroinvertebrates, for just all of the it was suitable for like just the geology and the ecology of that system. So like are there, I guess, well, like is this research mainly to inform sort of fisheries management or also like river restorations?
Initially, it was designed for both.
Mhm.
It was designed to evaluate the VSP metrics to determine how these populations were doing, to determine if we needed to do anything else, either from a hatchery perspective or a habitat perspective, because none of us knew how long it was going to take for the sediment to come through. In fact, I remember talking to Dave Montgomery during the ceremony for dam removal, and I asked him, how long do you think it's going to take for this stuff to wash through here?
Mhm.
A decade.
Mm.
No way, I can't believe it's going to be a decade. All the great research by Andy River, or Andy Ritchie, Amy East, John Warwick, and others had suggested that, yeah, most of it has gone through within that time period. George did analysis in his paper on Elwha, recovery of Chinook and steelhead, and basically he did a hydrology and sediment metric to look at the impacts of those two on Chinook salmon. And basically by 2019, he had concluded that most of the sediment impacts had pushed through the system. And that's very good, you know, very, very consistent with what the geomorphology studies suggested. That basically the first 2 to 3 years, a bulk of that sediment moved through the system, especially the fine sediment that we thought was going to be problematic. It moved through the system. I think only 10% of all the substrate was stored within the Elwha River main channel or floodplain channels. So the river was very efficient in pushing that stuff through.
Wow.
Also, a couple of recent papers that have come out looking at log jams, because that was one thing that none of us, you know, we knew large wood would come down once the dams were out there. They was blocking that. But we didn't have an idea of the magnitude. Everybody was shocked at how much small woody debris was scattered throughout the whole river. And like I say, a couple of recent studies have come out showing that log jams increased dramatically in the first couple of years after dam removal. And then they stabilized, and basically you have the same number of log jams now as you did then, but they've increased in size. And you've seen somewhat of a decrease. You also saw an increase in single logs that were stored along the river, but that has decreased over time. And their theory is that a lot of that wood is racking up on all of these log jams. So, so yeah, you know, I don't think there's any current projects planned right now. There are a couple, like the Lower Elwha tribe is really concerned that there's a barrier in Rika Canyon, which is in the park. We haven't seen any Chinook reds up there for quite a while. I would argue that we haven't seen the densities that we saw when we saw reds up there, so I don't think we're pushing fish up there. It's also within a park, so trying to do something with that is difficult because it's a wilderness area. And there's not a lot of agreement on, you know, some people think we got to go in and remove it. Other people's like, we get a big enough event, it's going to come out of there. My opinion is, look, as a non-geomorphologist, if you see successive pictures of that particular spot, it looks like a head cut that's moving upstream. And I think that head cut's gonna undercut these boulders that are causing the problem, and eventually they'll topple and we'll have fish passage. It's a dynamic system up there. I think, you know, it's probably always gone through this, and I'm sure it will recover. But to kind of try to get fish in the upper basin, they're talking about flying fish up into the upper basin.
Wow.
Chinook and coho. So to get them into areas that are not accessible currently.
Wow.
Flying or just airdropping?
And I think it would be more, I don't know, I haven't seen the plans, but my assumption is it will be more like what we did with bull trout prior to dam removal. Lake Mills, which is upstream of Glines Canyon Dam, was a real hot spot for bull trout. And so we caught fish, put them in fish totes, flew them up into the upper basin. We had crews up there. Basically, they would set the tote down, detach it, and then come back and get another tote. And the crews would move those fish into the river upstream.
It's like Mission Fish Impossible.
Yeah. Yeah, yeah, it would be.
Is there any major impact of the delta, all sediment went up to the, I don't know what you call the terms, that kind of form these sand bars,
Yeah, it did originally, so we saw
all disappeared now?
the delta really expanded during the first few years, and John Warwick has a couple, and Delph have a couple papers on this, but the delta basically it grew substantially. And to a point where we were concerned that it might become a barrier. We never did anything about it, but then I think it was probably 3 to 5 years afterwards, it started eroding. And so I think it's, you know, at the time they quit monitoring, I think it had slowed, but maybe had not reached equilibrium yet. But I think there's one thing that, folks don't talk about is that people out and working on the Elwha talk about it, but that all of that sediment may significantly stabilize the beaches to the east of the Elwha, because the primary drift is to the east. The city of Port Angeles they've had to riprap that is hooked to maintain it. And so all of this sediment will likely push through. And I don't know if anybody did any data collection that far to the east or not, but my guess is that has significantly helped the erosion along that beach.
All right. I think we don't have any other questions, but yeah, I think, thank you again, Dr. Roger Peters, for coming out.